Right. Um, so we'll talk a little bit about aseptic technique. So we talked a little bit last time about cell cultures and the importance of not having things contaminated. So aseptic technique is a technique that reduces contamination. So remember, A means without, and septic, right? A septic tank holds contaminated material, so without contamination, right? So aseptic is fundamental because if in a biotechnology lab, we have to continually sample things for purity, and if there are any contaminants, bacteria, cells that I don't want, then I have to throw away the whole batch. And a bioreactor full of stuff could be millions of dollars wasted. Right, so aseptic technique is really important. We'll do things like clamshelling. So if you look at this picture right here, this first picture on the top, right, she's holding this plate just a little bit open, right, so that things don't fall from above, right? It's called clamshelling. So we don't take lids open and we don't set them on counters, right? We crack them open and we work with them that way. You'll notice in this picture, the second picture, right, when we put fluids in or take fluids out, we do it at an angle, right? Again, so I'm not dropping things in my sample, right? I'm sliding things in to remove things from my sample, and it reduces contamination. Right, my hand is never over an opening, right? And then we'll use things called laminar flow hoods. So a laminar flow hood, what that'll do is it'll take air current from above, so there's air current coming down this way. That air current has gone through a HEPA filter, so it filters down to viral size. Right, so it's essentially just clean, sterile air, and it blows down, past, and out the opening. So this air is flowing down, and it's flowing out the opening here. And what that does is keeps any exhaled gases from the technician here from entering the hood, right? Or anything from people walking by entering the hood, right? We have bacteria floating and viruses floating, right? People sneeze, people cough, but it keeps all of that flowing out of the hood and doesn't contaminate. Again, you can see that she's pipetting from the side, right? So she doesn't contaminate her solution. So we have a whole bunch of things, but the important thing is that it takes conscious effort, right? Aseptic technique takes conscious effort. And we'll talk about that a little bit. So some of the tools we use, right? You need to know the tools. Inoculating loops. So a loop, these are the ones we use in lab, if we were actually in lab right now, right? Some type of a smear loop, inoculation wand. It's a simple tool for retrieving bacteria, right, in culture. So if I had a petri dish and I had a colony in a petri dish, I'd use a loop to remove the colony, right? Bacterial spreaders. Over here, they kind of look like hockey sticks, right? Is used to spread evenly on a plate. So if I had a petri dish here, right, and I had my medium here. So when we looked at before, if I put my 100 microliters of sample, right, in the middle, I could use a spreader to spread it evenly across the dish. But when we're doing our serial dilutions, we talked about those, that's how they would have spread it. And a needle, right, if you look at the other side of this, is a needle. And the needle is used for creating stab cultures or deep tubes, right? And that's why I'll stab it in. And I'll have another video I'll post where I'll show you how to use all these. So, aseptic technique. So the important thing is that you need to use sterile equipment. Sterile equipment is done by heating. So I'll show you a video. It's actually the video associated with this picture right here. So I'll show you that too. But things that you need to know, so come back to this, that you should heat the loop until it turns red, right? And then you must let it cool before you touch your bacteria so you don't cook them. But I'll show you how to test that with the agar. So the loop should be in the dominant hand, and then pass over the blue cone for 10 to 20 seconds. Once you start, you can't put the loop down because then it's contaminated. You have to start, right? So all of this right here, right, will be covered in the video. And I'll show you that new video, but I'll post this so you can come back to it. Components, we talked about always clean environments. We're going to use 10% bleach in our class, so I'm going to do that before and after every procedure. Right? We're going to wear PPE, so we're going to wear gloves at the start of every task. We're going to cap and recap. Right? We're not going to leave caps off of plates, off of reagent bottles, off of anything. Because when you leave caps off, you contaminate things. Right? We're not going to set caps down. So when we take a cap off, we're going to hold it in our hand until we put it back on. Right? Remember, lids of culture plates should not be fully removed. They should be clamshelled. Right? And the clamshelling, remember, reduces contamination. Right? Like he's pouring agar here, right? But he's clamshelling as he pours. He didn't take all the lids off and set them on the counter and then put them all back. Right? This person over here is removing a lid, and they're holding it between these two fingers they're not using so they can put it right back on. Right? We don't agitate broth if we don't have to. Right? If I shake things, I make aerosols, and aerosols bring contamination. And this should be tilted away from burners. Right? If I have a Bunsen burner, it creates a convection current, right? And if I had my lid off this plate here at this bottom picture, then any bacteria in the air here would be carried by the convection current into my plate. So I'm going to try to clamshell away from Bunsen burners if possible. Then properly labeling, right? So things that should be done, properly labeling. So if I look at this picture here at the top, I should be labeling around the outside of the plate, right? I should be labeling in the middle. By the way, this is fungus right here. This is contamination of that plate. We've got the bacteria, we've got the fungus, but it says TSA, right? So that's from the culturing of like an airport, right? Which is kind of interesting. So they're missing some stuff here on this labeling, but we should label. So when we make plates in class, we'll label with the type of agar, so we might say LV. Any additives, we might say there's ampicillin here. The date poured, so we can say this is 10/7, and initials. Right, so initials that we poured it. So when I make my plates, I should label them. And then when I culture, I should continue my label on there. So the organism, if I'm culturing E. coli, I should write that. Right. The plate, this is number 1 of 5, right, if it's a series. And again, my initial and date. If I'm doing this tomorrow, it'll be 10/8. Right, slants and broth tube, same way. I'll do it on the edge of the tube, right? But I don't want to write on top of what I want to see. Right, so here's an example of what not to do. Right, so if you look at this labeling here, you'll notice the labeling at the top is on the bottom of the plate. So I want to label the bottom and not the lid because lids can be changed. But if you look at here, I've got all these, right? Like I've got polio, and bubonic plague, and influenza, and yellow fever, and cytomegalovirus, and respiratory syncytial virus. So all these really terrible things, but it's all labeled on the lid. But there's also a lot of other stuff missing from the lid. Right? So I don't want to label lids because lids can get mixed up. Right? So this is kind of a quick outline, but go ahead and watch the other safety video, and then you'll have a better idea. And hopefully we get back in lab soon. All right, bye.